Okay, let's have a class. We're going to talk about lacto-operon today. It's a very comprehensive concept. It's a little twisted to your mind. But let's just go over it, because when we talk about lacto-operon, you get to review lots of the material we have been talked already. And also that's a good material for reviewing the exam 3. Okay, so before we go, just want something on the e-campus. I want you to pay attention. So the exam will be November 18th, I already said, and Thursday we will do a review. And then I have 3 sets of the practice exam questions, which is exam 3, practice 1, 2, 3 on the e-campus. Practice 3 basically is very similar to the 1. But 1 and 2 absolutely is very meaningful, so we should do it. Then I also in here had a blackboard which is talk details about the aerobic respiration, the calculating the ATP and all kinds of the chemical name and the structure there. And this is for you to study and get extra points. And I also have a video recording here, which is, you can see, very detailed. We talk about all the side chain of the amino acids. And I also have another video which is talk about the ATP calculation. So these you should watch if you are unfamiliar with or you prepare for the exam 3. So I put them back from my previous years of the recorded, and then you could use it. Okay? So these are the things, just remind you. You should have to study very well with all these material to help you to do good in the exam 3. Okay, so let's talk about today's work, which is lacto-operon. It's a comprehensive concept, but let's talk about these one by one. The outline of this class, of this exam 3, is we talk about is DNA replication, going to RNA, we call transcription, and then going to protein, first with amino acid, then with protein, we call it a translation. We already talked about all these material. Now, I'm going to skip these slides, but you get to know. It could happen is RNA back to DNA if there is reverse transcriptase. This is discovered by Temming and Baltimore in 1970. And it's already explained to you very well how to do it. So in the old time, we have reverse transcriptase PCR. I'll just skip that because it's not difficult to understand. So I'll just let you know it's going to happen. But we want to focus on here. So this is for bacteria, their life cycle. Now, for bacteria to survive in their normal life, for their normal activity, all kinds of the physical activity, their gene to regulate that, going through replication, transcription, translation, we call those a housekeeping gene. Or we call those a constitutional gene. However, regarding the nutrients taking, what kind of the nutrients they're going to use, especially what type of the sugars they're going to use, they have their precisely gene regulation corresponding to that. The reason is bacteria's lifetime is very short. In their very short lifetime, they have to using the nutrients very economically. That's why we have a different gene regulation for that. If it happens from DNA to RNA, we call it post-transcriptional regulation. If it happens from RNA to protein, we call it post translation regulation. And in the scientific area, their studies in microbiology or bacterial physiology, we got lots of the information. All we say we will study for the post-transcriptional regulation compared to the post-translation regulation. Therefore, you get lac operon, you get streptooperon, you get maltose operon, you get arabinose operon, on and on. And we can talk about that the whole day, because we will study that. And we lots of them, we're using E. coli as a demonstration, as a demo. So if we want to look at the overview picture, you have a nutrients, and we call it a stimulant. And the bacteria in their cell wall system, they have a sensor. So the sensor will be feeling about it. Once the sensor feeling about it, it will be amplified the signal, which means you have something going on, I'm feeling about it, I'm going to enlarge it to let you be feeling better. And then we have gene regulation corresponding to that. Now these nutrients typically is lactooperon, lactose, tryptophan, maltose, all those type of sugar or amino acids or fatty acids, all those macronutrients, nutrients molecule. So this is the overview picture, what it looks like, why it's being, we talk about these things. Now before we talk about the lactooperon, there are two basic knowledge we need to know. Is we learn in your biochemistry class or genetics class, we call it a positive control, negative control. What that really means, then inducible, repressible, all those things looks like confusing. What this means, this is we call it a structural gene and a functional gene. So let's say I have a gene which is linear. Most of the bacteria is linear. I'm talking about is only 3 positions at the beginning, regardless how complicated it is. And I put them in a linear order, which is A, P, O. Okay, that's we start from talking about here. And I want to be cautious is that in the real structure, it's not an order like that. It sometimes is alternate or interacted with each other. But when we explain to you, we put them linear. What is A means? That is activator binding protein. What is P means? That's promoter. And we already talked. What is O means? Operator. So first of all, you need to know what we already talked. So let's do a very brief review. Promoter is an important structure because it will be combined with an enzyme called RNA polymerase. Now, by the way, the RNA polymerase, remember you have alpha 2, beta prime, omega, and sigma factor. And you have a core enzyme, you have like a coenzyme. That's, you can go ahead to do the review. Now, what is a promoter? Do you remember we talked about the +1, -10, -35, this TA, AT, TT, GA, CA. Remember? This whole area is a promoter. So, eventually what they are regulated for is to see whether RNA polymerase is combined with protein or not. Because if they connected with each other, then RNA transcription will happen. Now, of course, the transcription is always from 5' to 3'. If they are not connected with each other, then the transcription will not happen. In other words, of course, if the transcription will not happen, the translation will not happen. So this is what they are regulated for. So, regardless of what they do, they regulate this guy. Okay? Now, since you have a left side, activate the binding protein, we call it A, and you have a right side, it's called operator. Everything happened when you're facing the blackboard on the right side. This is called a negative control. And everything happened on the left side is positive control. This is the first thing you need to know. Now, what are here? This is functional gene. Now, typically in lateral operon is lac Y, lac Z, and lac A. This is we're going to talk later. So first of all, you need to know right side is negative control, left side is positive control. Now, negative control, there are two different categories. Right there you can see. It's going to be negative control with inducible gene or with repressible gene. And the positive control also have two categories. It's very similar, which is inducible and repressible. Okay, how you understand about it? What means inducible? Inducible, which means originally there is a repressor, and I induce it. I let them not become a repressor. The mechanism is what we already talked about, is allosteric regulation for the enzyme. Now, what is a repressible? Repressible, we will let the repression, which means inhibition, more strong. So this is basically is a co-repressor. So these are the basic things we want to talk about before we go. Now, we want to give you some of the examples. The examples is already on the blackboard, so I want to emphasize. Let's talk about negative control with inducible gene. So what this means? Okay, let's very simple talk about these things. A, P, O. What means the inducible gene? Originally, they are something which is called a repressor, and you can understand it's an inhibitor. This is equal inhibitor. The inhibitor, we call it a repressor, is attaching to the O, attaching to the operator. Because they attach to it, therefore, the RNA polymerase cannot attach to the P. Therefore, the transcription will not happen. This is called originally what is called a repressor. Now what happened is there is some chemical. We call it an inducer. So let's say originally it looks like this. And I add an inducer there. When I add an inducer, the morphology is going to change. It maybe become like this. And then because the morphology has been changed, it's not going to attach to the O. And what happens is RNA polymerase attaching to the promoter, therefore, the transcription happens. At this moment, the red color here As a blue color here, we call it an inducer. Now, what is a good example of this guy? Is a lacto operon. You're going to talk real quick. Okay, now, since this example for inducible gene, how about repressible? Repressible, which means still APO3 position. I had a repressor which is right here. Okay, that's a repressor. It doesn't do anything, it just stays in the cell membrane system. It doesn't do anything, it's not attached to the O. Okay, so RNA polymerase attaching to the promoter and the transcription happens. Very good. However, I have something comes. This is called co-repressor. Once the co-repressor attached to the repressor, they change the morphology, they go ahead to attach to the O position, which is attached to the operator. Therefore, RNA polymerase cannot attach to the promoter and the transcription cannot happen. At this moment, this guy we call it a co-repressor. So we call it repressible gene. Now, what is the example for this one? Is tryptophan operon. We will talk briefly after we finish talk about the lacto operon. Okay, this is for negative control. Now, what is positive control? I'm going to write here. Okay, positive control. We mentioned about negative control happened on the right side at O. Where is the positive control going to happen? Obviously, it happens something at A. So, what's the story? I have something there. Originally, it's just there. And we call it activator protein. They just stay there. And one day, I have some chemical goes in. It's called the inducer. Once the inducer attached to the activator protein, they go ahead to attach to the A. And they help or prompt RNA polymerase attached to the P, transcription happens. What is the example of this one? Maltose operon. I'm not going to get to this. This is usually people talk about microbial physiology. They're going to talk this one, which means bacteria use maltose is not that easy. It also has some regulation for it. So that's called a positive inducible gene. What is a repressible means? Which means originally I have something attached to here. That's activator protein. And the RNA polymerase attached to the promoter, transcription happened. However, one day, I have some very naughty chemical there. And that is called a repressor. And they change the morphology of the activated protein. They don't let you attach to it. And they also prompt the RNA polymerase attached to the promoter. This transcription not happen. Okay, so this is a for example, what is we call negative control with the inducible gene, with repressible gene, and positive control with inducible and repressible. It sounds like a little difficult. We have slides for this. So you can well understand about that. And I also tell you, the new edition of the textbook released, deleted these figures. this is an older version, about 20 years ago, the textbook from my work here. They have all these 8 pictures, which is very good for your understanding, but the new textbook already deleted that. That's one of the reasons I never ask you to buy textbook, because they have a new edition, they have to do some change, the price is increased, skyrocketing, and lots of the good information they deleted because they think you're smart. That's what happened. Okay, but these pictures I still left it. It's really good to explain to you the positive and negative control. Now, what is the thing makes you difficult? Is in the real life, we have is a demo. We have a model. But the bacteria leaving their environment, it's not that simple. Lots of the times, they are controlled by both positive and negative control. Both inducible or repressible. That is why it makes the story more tricky and more difficult. And we're going to talk about the lacto-operon, which can demonstrate that what they talk about. Okay? So let's talk about the lacto-operon. So I have to remove this whole thing, and basically this is going to be, the information here is reviewing what we talk about, the transcription, and also the 2 slides in this section of the class. Then we talk about the lacto-operon. Okay, lacto-operon. Lacto-operon, you could understanding, is the research outcomes for E. coli, how they using lactose. It is a very well studied and established model. The outcome is coming from Menard and Jacob. Menard is Jacqueline Menard and Francisco Jacob. Those are 2 well-known French biochemists and microbiologists. And Jacob is Menard's students. They are in 1950s published the outcome of a lacto-operon and win the Nobel Prize around the 1960s to 70s. I forgot the detailed time. That is the reason why in your biochemistry class, in your genetics class, even your nutrition class, we talk about the lacto-operon. I know we talk about the lactose for other things. It's like it's a disaccharide, it's like a lactose intolerance, all kinds of things. But that's what it is studied very well. Okay. So first of all, we have 2 preparing basic knowledge you need to know. What is lacto-operon looks like? What is the structure looks like? I'll make it easy. I don't want to make it very complicated. It's C, P, O. What is C? Catabolite activate binding protein. We sometimes call it cap. What is O? This is promoter. We already mentioned. It should be connected to the RNA polymerase. What is O? Operator. And O, they separate into 3 sections. If you want to talk about this, there is O1, O2, and O3. O1 is very important. Now, this is their structure gene, and their functional gene is right here. There are 3 major functional genes, which is we call Like Z, let me think. Like Z, like Y, and like A. Like Z, like Y, and like A. What means functional gene? Functional gene, each of them responsible for one of the function, which means they're going to do something, not just stay there. What is like Z? This is beta galactosidase. Beta galactosidase basically is breakdown. Lactose become glucose and galactose. Well, I don't have that galactose, yes. What is lac Y? This is lactopermease. What is permease? Permit. They let you come in. What is lac A? This is a transacylase, but the function is unknown. So, if you want to say very simple, what is lac Z? Beta galactosidase. Break down lactose, become glucose and galactose. What is lac Y? It is lactopermease. They let the lactose go inside of the cell membrane system. What called permease? It's permeate or penetrate, go inside. Lac A, you can say it's unknown. We don't know what's the function yet. Okay, so that is the basic structure. Okay, now we want to talk about what happened. This is the bacterial cell membrane system or cell wall system. Okay, I'm going to dig a hole. So, lactose can go inside. Lactose go inside. They rely on an enzyme. They're not going to just go in naturally. Every function have the enzyme to regulate that. What's the enzyme? Lactopermease. So they rely on lac Y is lactopermease. Now we want to talk about what is the structure of lactose. Okay, what's the structure of lactose? What are the lactose combined? Is glucose and galactose. What are they? What are they looks like? You should be very familiar with what it looks like. Okay? H, H, OH, OH, H, OH, H, H, CH2, OH. What is that? That's glucose. Okay, what is galactose? Exactly the same. There is only one exception. So we draw like that. Exactly the same, we can just copy. And you can using this opportunity to study the extra points of the exam 3. And I said exactly the same. There is only one difference. What are the difference? Carbon number 1, carbon number 2, carbon number 3, carbon number 4. The only difference is OH and H upside down compared to glucose. Okay, so OH is on the top, H is at the bottom. That is galactose. Okay, how they connected? They connected right here. I'm just trying to simple. Based on 1,4 glycosidic bond. Because for glucose, this is 1 prime. For galactose, this is 4 prime. So 1,4 glycosidic bonds. That is the structure of lactose. Okay, if lactose has beta galactosidase, what are they going to do? They're going to separate it. They become glucose and galactose. Okay, this is normally happen. But in the bacterial cell wall, the research has been showing, although 99% of the time is happened this reaction, they also had a side reaction will happen is beta glycosidase instead of hydrolyze lactose become glucose and galactose, they change the glycosidic bond position, become a side products. What it looks like. This is the glucose. Okay. Wait a second. So this is the glucose. There are no question about that. That's a glucose. And we have a galactose. Okay, OH, H, H, OH, OH, H, OH, H. Instead of happened here, their reaction happens goes here. So they forming 1,6 glycosidic bonds. What is this called? Allolactose. This side reaction is important. Okay. This is the two basic knowledge you need to know before we go to the twisted mind explanation. Okay? So, say it very simple. You have CPO, like Z, like Y, like A, each have their function. And we did the picture for you how the lactose go inside of the cell membrane system, rely on the enzyme is lactopermease, and the reaction happened is beta glycosidase. Glycosidase has a main function, break down lactose, become glucose and galactose. However, it's very important for the side reaction, they have a allolactose will be generated. So, then we go back, talk about what happened to the lacto-operon. Okay, let's draw one more time. Remember CPO, lac Y, lac Z, and lac A. Okay, the scenario right now is number one. If there is no lactose, what will happen? If in the environment there is no lactose, there is something will be attaching to the O. We call it a repressor or an inhibitor. Okay, there's a repressor. attach to the O. Once they attach to the O, AEM polymerase will not attach to P, the promoter. Therefore, transcription will not happen. Which means this reaction will not happen. That sounds very easy because there's no lactose. Obviously, you're not going to happen this guy here. Now, how it works? This repressor, it is a helixy structure. And they connected to the O. And remember what I said. This linear structure is just to let you understand easily. In the real life, it's not like that. It's like this. The O is like this. The operator is looks like this. And then they separate to become O1, O1, O2, and big chunk of O3. And how the repressor they go there, the inhibitor? They are attaching to the O1 position. So the operator has 3 positions. O1 is very important because that's what really the position. The tetramer, the repressor goes there, and this is a tetramer. That's how they work. There's no lactose. Okay. If there is a lactose, what is going to happen? If there is a lactose, the lactose is going to go in, glycosidase is going to be, beta-glycosidase is going to do something, and they will generate allolactose. And what is the allolactose looks like? The allolactose is right here. Once the allolactose connected to the tetramer, the morphology will be changing. Maybe become this. Okay, if there is allolactose. If the morphology has been changed, obviously they're not going to attach to the O. Okay, if this is not attached, what happened? AEM polymerase will attach into the P. They're going to attach with each other, and the transcription will happen. If the transcription happens, what this means? Bacteria activate beta-glycosidase. This reaction will happen. Then they use lactose become glucose and galactose. At the same time, little bit of side reaction for the allolactose, but that's why they can use it. So at this moment, we call allolactose is an inducer. And originally the tetramer is a repressor. Once they combine with the inducer, the morphology changed. So we call it an inducible gene. That's why we call it an inducible, repressible, negative control. Okay? That sounds very straightforward. It's very simple. But which one we haven't talked? This guy we haven't talked is C, right here. Remember, we talk everything is on the right side. We don't have to talk about C yet, and C is important for the cat. So we're going to talk about that. This is easy. Okay, no lactose, lactose present, allolactose. Now, at the beginning, Menard and Jacob, when they do the research by themselves, they only stop here. But what we call research, People need to repeat it to do it. And sometimes when during the replication, they also have a new discovery, new material found. What they find is very interesting. This is what we're going to go back to talk about the growth curve. How to draw the growth curve. Y-axis is the time. Y-axis is the population of the bacterial cells. And what happened? They think about, in a very occasional moment, they are doing the research to let the E. coli grow in the media which is containing lactose and glucose. And what they find, the results is very interesting. They find E. coli has a secondary growth curve. And then they further to do the research. They find with the coexistence of lactose and glucose, bacteria use glucose first. Then they use lactose. This phenomenon called the dioxy growth. This is some side reaction. When they do the research for lacto-operon, they find it. We call it biphasic growth. It's very interesting. Now what happened? It's related to here. Which means bacteria to use lactose. Which means activate lacto-operon, they need another presumption is in the media there is no glucose. If there is a glucose, they're going to use the glucose first. Once they use the glucose, then they use lactose. That totally makes sense. Why? Glucose is a monosaccharide, and the lactose is a disaccharide. And we of course use monosaccharides first. This is very simple understanding. In the old time, we only have cash. You have $5 bill and $10 bill. Of course, you use $5 bill first. Because otherwise, you use $10 bill, you have two $5 bills and have lots of the pennies and the coins. It's make your pocket looks like stuffed. So we don't like that. It's same thing for the bacteria. It's a very economical way to handle the situation. Okay? So we said it very simple. More and more research has been showing. Bacteria activate lactose, operon. They need lactose. They also has a requirement, there's no glucose. Now why? This is need going to see what we already talked. Okay. Let's draw here. You still remember the basic structure of a bacterial cell wall membrane system. That's called fluid mosaic model. Okay, what it looks like is lipids, phospholipids, across the cell membrane system and interacted or inserted with protein. So there's some protein structure there. Okay. Let's say ABC, okay? That's a cell membrane system. Okay, I have a glucose coming. If there's a glucose go to the bacterial cell wall, cell membrane system, how do you do it? This is go back to see when we talk about glycolysis. Glucose become glucose 6-phosphate. Okay, let's do a review what it looks like. That's a glucose we already draw millions of times. So I'm helping you to review right now, okay? You should be following me to draw on your notebook. That's a glucose. What is glucose 6-phosphate? Okay. Exactly the same. That's why I said if you get one, you get all of them, or a bunch of them. That's glucose 6-phosphate. Okay, how they come in? When I said in your biochemistry class, we talk about human being and animals. So the enzyme called hexokinase. But I said this is a bacteria, microbiology class. We focus on the bacteria. How they do it? They rely on group translocation. Now how they do you remember? They rely on phosphoenolpyruvate. They go this. They go here, what they needed? They need an enzyme, complex 1. But before that, they need heat stable protein. So we just say HR. Okay, heat stable protein. And they go here, enzyme complex 1, then go here, enzyme complex 2, then go ABC, and then they generate this guy. Now why they do this way? It's more easy to do because when the glucose goes inside the cell membrane, it's already become glucose 6-phosphates. It's speed up, it's going through glycolysis. Remember, this is the reason you have glucose. How about if you don't have glucose? These things still will happen. But they don't go this way. They'll activate a chemical here. Activate an enzyme here called AC. What is that? Adenine cyclase. Okay, what is adenine cyclase? The function will be ATP rely on AC, adenine cyclase, will become CAMP. What is the reaction looks like? Extra points, another 5 points, so you need to know how to draw it. Is that right? How to draw it? We help you review. NH2, N, here, N, here. So what we do? Our phosphates go here. Then what we do? H, OH, H, CH2. Then go here. Phosphate, sugar, phosphate. Go O, sugar, phosphate. Then go here. Sugar, phosphates. What is this called? That's ATP. Okay, if I had cyclase, what will happen? There happens something around here. So what will be? All the others are exactly the same. You don't have to worry. Okay, let's draw one more time. I like to draw these chemicals. It's really good. You know, once you draw, you always remember N, O here, O here, O here, H, OH, H, OH. That is, OH, H, it's okay, doesn't matter. There's something going to happen. What are we going to happen? Right here. Go back. What this called? CAMP. And I said, most of the time, CAMP is not good. Exam 2, when we talk about all these diseases, most of the time the mucus comes out, is ATP becomes CAMP. Like the money has been packaged in your wallet, you can locked in your wallet, you cannot use it. With one exception, here is really good. Because lactose operon, when they start to do, they need CAMP attached to it. But what is the presumption of CAMP attached to the C place? Or we say it's an A place. Is no glucose. Or very limited glucose. Because with very limited or no glucose, they didn't go this way. They go this reaction. Okay, that is pretty much the whole story of the lacto operon. I'm going to give you a conclusion table. But before that, I want to mention one thing. When I was a student, I like to ask a question. Around 25 years ago, I asked my instructor, he's like 82 years old in China, in animal biochemistry class. Basically, his biochemistry class make it a little bit easy, so we call it animal biochemistry class. Did you notice that? We talk about lactose going, then generate lactose, glucose, allolactose. Where is the first allolactose comes? Did you ever think about it? And you say, oh, you need these activated, then allolactose coming. But where did the first one goes? This has not been coming. Is that right? The lactose has not been coming, has not been used. How come you have allolactose? Well, my advisor told me, this is the same question like egg and chicken, which comes first. I think this just make a joke and teasing me. So when I become an instructor in the university 12 years ago, I asked the question about 25 years ago. He said it's like a chicken and egg, which comes first. And I looked at the literature, I looked some textbook. The best answer is that in the bacteria life cycle, they have a minimal trace amount of the allolactose is ready to use. That's why here, we do not really say there is no glucose. We just say very limited glucose. Because even on this bench, if you do nutrition or nutrients analysis, I better say there's a glucose there, very few, very limited. It's, of course, cannot compare to the glucose in our bacterial media. So, how they started earlier, the best answer is, in the bacterial cell membrane system, they more or less has a trace amount of the allolactose is ready to initiate it. That is the best answer. This is going to be the trace amount existing in the bacteria. Okay, so this is the story. Now, let's do a conclusion table. We talk about lactose operon. And we said lactose operon is correlated and connected with the presence and absence of the glucose. So therefore, if you do the math, there are 4 different situations will happen. 2 by 2, is that right? So 4 different situations will happen. Little twisted your mind, but let's talk about that. Let's draw 4 situations. Okay, I skip here. I just say lack. Okay? C, P, O. We draw C, P, O. It's just lack. Okay, go here. Lack, C, P, O. C, P, O. We already talked about. With presumption of the presence, absence of the glucose and lactose, there are 4 different scenarios. First scenario. There is lactose, but no glucose. Okay, what will happen? At this moment, the tetramer is connected to allolactose. Change the morphology so they do not attach to operator because no glucose, so they don't go to the direction happen glucose 6-phosphates. Instead, they generate CAMP with a very good positive and negative control. RNA polymerase attached to the promoter, transcription happens. That's the first one. Okay, second situation. We have lactose. We also have glucose. Okay, what happened? On the right side for the O, like we just mentioned, the tetramer, the repressor, is attached with allolactose, which is the inducer, so they do not connect it to the O. However, because there is a glucose, so CAMP is not generated. Instead, they have glucose 6-phosphates going through glycolysis. This would happen. Bacteria use glucose first. So, does RNA polymerase connected with the promoter? No. Does the transcription happen? No. Okay. Scenario number 3. There is no lactose, but there is glucose. What will happen? Because no lactose, the tetramer is connected to operator, sticks there, especially the O1 position. And with glucose, what happened? The CAMP not going to attach to the capside. Obviously, left and right doesn't work at all. So, does RNA polymerase attach to the PE? Of course not. Does it happen, the transcription? Of course not. What this means? This means you want to buy a house. And you talk to your parents, you talk to your mother-in-law or father-in-law looking for money, nobody gave you money. Nobody wants you to buy the house. Or you can simply say that you want to get married to the girl. You parents and your mother-in-law, father-in-law doesn't agree with it. I don't know what you do here. In China, we have to get approved by both parents. That's why I always use an example to explain the situation with lactal operon. What is called a positive control, what is called negative control, is like the young couple is getting controlled with parents and their father and mother-in-law. Okay, which is called positive and negative control. So that's what happened. Okay, next situation. There is no lactose and there is no glucose. Okay, no lactose. Tetramer attached to the O position very well. No glucose, that's good. CAMP goes. However, we mentioned you have to be both working very well. Does this happen? Of course not. And this RNA polymerase is not reacted to the P. Okay. This will be in your exam 3. I don't need you to talk about all these. Extra points. Extra points for structure. Think about it. How many extra points we already mentioned? ATP, glucose, glucose 6-phosphates. All these things we talked about. Glycolysis we mentioned. But at least the conclusion you should know. Okay? Story behind it, it's up to you. This slide is extremely important. Okay? Okay, we have a couple of slides left, and you just do a self-learning study. I'm not gonna do this anymore because it's a little bit difficult. This is talk about the restriction enzyme, and there are certain recognized sticky ends, and then after do the gene cloning, and then they go to plated onto agar plates. Will be showing you the mutation or not with a vector. I no longer really talk about that anymore because it's a little bit difficult. I don't think you learned this in your genetics class, and I usually talk about that. 150 students, maybe like 10% will understand. So I don't no longer require you with that. You can take a look. I believe you learn in your other classes. It's a very interesting results, but we not gonna talk about that. The reason is, this is we talk about how they control lag Z, lag Y, lag Z, and lag A, not lag A, how they control here. They're using the restriction enzyme to cut a certain sequence, and they can reconstruct it. It's more genetics, so we not gonna talk about that. So the requirements here, you can skip these couple of slides. You can do a self-learning by yourself. But lactal operon, very important is this slide, you need to know, and this one you need to know. Okay, sorry. This slide you need to know. What is called dioxic growth, and this conclusion slides you need to know. Okay, what's the scenario will happen? If with or without lactal operon, what happens? Okay? So go ahead to do. You practice the exam. The best way come into the review section is you have some reaction. Okay, you're not going to just listen to me. Oh, A is not good. B, oh, C is not good. You well prepared. Look at your notes. Do those questions by yourself with your friend in the Evansdale library. Do it first. Then Thursday when you come in, you get well prepared to do the review. That's more effective compared you just listen to tell you the answer. Okay, that's what we always want you to do. Okay.